Thank you very much for agreeing to give a Build a Cell seminar, and you can get started.
Great. Well, good morning, everybody. I'm Jeanne Stachowiak here at the University of Texas at Austin. My lab is interested in biological membranes, their biophysics, how they're organized, and also how we can use them to build minimal systems. So I'll share some of that with you today. You're welcome to interrupt with questions. You can use the chat. You can also just speak up if the meeting allows that, which I think it does. And if you have trouble hearing me at all or something, please let me know. Okay, so before I start, I'm just going to acknowledge my group, and I also apologize in advance. I have this cold I'm getting over, and it's making me cough a lot, but I'm armed with lozenges and water, so I think we will survive it. But here's my group. Obviously, everything I'm going to share with you was done by them. I'm super proud of them. I will try to acknowledge their specific contributions as we go along. Okay, let's see. Yes. Okay, so in our lab, we're interested in biological membranes, as I've already told you, and we kind of study them from this reverse engineering approach, which I think is relevant to this build a cell community. So we think of the cell like a complicated system. And like other complicated systems, we can learn about them by taking them apart and looking at all the components that comprise them. In our case, that's the lipids and the proteins, the glycosylation, everything that's going on at the membrane surface. And then ultimately, after studying components, we often try to put back together some kind of minimal system. Now, traditionally, reverse engineering, you think of trying to make an improvement upon the initial system. But here I've shown this little toy card to indicate that we're not really so much trying to improve upon cellular membranes. Our goal is more to make a system that we can control. And that control is useful for two reasons. One reason is if we're trying to test hypotheses about how a cellular process works, having a system with a minimal number of components often makes it easier to isolate very specific questions. Then on the other side, a minimal system can be useful for therapeutic intervention, for modeling some aspect of the cell in an artificial tissue, very much in line with that build a cell mentality. Well, let me just check the chat. It looks like there's something. Okay, yes, all good. All right, so with that in mind, there are several questions we're going to tackle today. The first one is how are membranes organized and shaped very much at the fundamental level. Then we're going to take some of those ideas about membrane organization and shape and ask how they impact processes in the cell. And then ultimately, we're going to end with some more applied work, thinking how can we harness some of those ideas for therapeutic delivery. Okay, so we'll start with the super fundamental stuff. How are membranes organized and shaped? Excuse me. So the first thing I want you to think about and understand is that curvature, high curvatures of the order of tens to at most maybe 100 nanometer radii of curvature are important to the function of really every membrane that we find in the cell, whether it's the organelles or the plasma membrane. At the plasma membrane, it's obvious how curvature is so important. It's important for importing and exporting material. For example, envelope viruses assemble at the plasma membrane to export material, right? Other processes also export material. Then endocytic structures like caveoli, endocytic vesicles, which we'll hear more about in the second part of the talk, they're important for importing material. So this curvature is essential to both export and import. So biophysicists and cell biologists have been interested for a long time in how proteins and lipids work together to bring about curvature. And certainly, if we're thinking about building a synthetic cell, having a way to import and export material is one of the key steps, right, to building something that's more cell-like. So we're interested in that question too. So there are two major ideas that are popular if you search the literature for how do proteins bend membranes, and they both involve structure. So probably the oldest idea is that you have proteins that have a very specific curved membrane binding interface. Here I'm showing this bar domain. There's a whole family of bar domains, but there are many other proteins that fit this description. They basically act as scaffolds. They imprint their curvature on the membrane, force the membrane to take on their curvature. So that's one idea. A second idea that's almost as popular is the idea of insertion. So there are proteins that insert usually a helix, sometimes a loop, into the membrane. And if that helix is amphipathic or has a hydrophobic face, then it acts like a little wedge and increases the surface area of one leaflet of the membrane relative to the other. And that also can be a way to generate curvature. So as I said, both of these ideas are very much driven by specific structural motifs. And that has been a really useful kind of paradigm for searching the proteome for proteins that could be involved in shaping membranes, but it's incomplete for the following reason. And that is that we're now recognizing that many of the proteins involved in curved membrane structures, like endocytic vesicles, for example, viruses, many of these proteins have substantial regions of intrinsic disorder. So here we're looking at a protein called epsin. It's one of about 3-4 dozen proteins involved in clathrin-mediated endocytosis, which we'll hear about later. And you can see that it has a large intrinsically disordered C-terminal domain. The N-terminal domain is well folded and actually is inserting a helix into the membrane, which is the mechanism we were just talking about. But you can see, of course, the C-terminus is disordered and therefore has a large hydrodynamic radius, acts more like a polymer than it does like a well-folded protein. So as you might imagine, if you take these kind of bulky polymeric-like domains and you crowd them together on the membrane surface, they're going to repel one another. And in this case, this disordered domain has a high net negative charge, so it tends to be repulsive toward copies of itself. And that sort of steric electrostatic repulsion can create a pressure on the membrane surface. And one way to relieve that pressure is to bend the membrane, as the cartoon there is showing. And we've observed that in a lot of our experiments. So here we're looking at giant vesicles. So we're going to spend a lot of time today talking about these giant vesicles, which have diameters of the micrometer range. And since this is the build-a-cell, I'm sure a lot of you are very aware of giant vesicles. But this is a confocal slice through the giant vesicle. And you can see that when a protein binds the outer surface of it, it causes this bending, generates all these tubules. So work like this in our lab over the past decade or so has established that if you take disordered proteins, which are bulky and repel one another, you crowd them together on the membrane, they generate a steric electrostatic pressure that drives the membrane to bend outward. It gives you convex membrane bending. So that's sort of some background. But the first topic I'm going to share with you today is what happens if the opposite is true? What happens if instead we look at disordered proteins at membrane surfaces that attract one another? Will that have any impact on curvature? If so, what kind of impact? So as I'm sure many of you are aware, attractive interactions between disordered proteins have been getting a lot of attention lately. It's increasingly clear that when you have disordered proteins that have some multivalent attractive interaction with one another, often also including structure domains, nucleotides, that you can set up these liquid-like, flexible, disordered protein networks when there's no membrane around and these networks just form in solution or in the cytosol. You get these big liquid-like spherical droplets. And droplets like this have been found to play a role in many different pathways in the cell, from organizing the genome to compartmentalizing reactions. It just seems like every week some new function is assigned to these liquid-like protein droplets made of disordered proteins. And exciting for us, since we're excited about membranes, is that increasingly these protein phase separation or liquid-like protein condensation is thought to play a role in membrane processes. For example, the immunological synapse, focal adhesions, cell-cell junctions, now all thought to rely to some degree on this liquid-like assembly of disordered proteins. So with that as the motivation, we're going to ask this question. Could protein phase separation, liquid-like assembly, attractive interactions between disordered proteins, could that have any impact on membrane shape or curvature? Okay, so this is the work of a graduate student in our lab, Fong Yuan, and Fong took a really fundamental approach to this question. He took a very well-studied disordered protein that's known to form liquid-like condensates, the FUS-LC domain, and concentrated it using nickel histidine chemistry on membrane surfaces to see whether this protein condensation on a membrane surface can drive any change in membrane shape. So the FUS-LC protein, there's only two things you need to know about it. one, I have already told you, it forms liquid-like condensates. It wants to form into these liquid-like networks. And then the second thing you need to know is that because of the specific amino acid composition of FUS-LC, the higher the salt concentration, the less soluble this protein becomes in the medium and the more it wants to phase separate. So we will use the salt concentration as a knob we can turn in these experiments. Okay, so this is Fong, and this is his work. So Fong, again, taking these giant vesicles, he attracted this FUS-LC protein to their surfaces, and you can see right away that the protein phase separates into this sort of protein-rich region and a protein-depleted region. And in 3D, if you project this thing, it looks like sort of a cap sitting on the spherical surface of this vesicle. Interestingly, these protein-rich regions are not stable. They quickly degenerate into these tubules that start at the membrane surface and penetrate inward. And the tubules are really interesting. They're floppy and flexible, suggesting that the protein network is not rigid. And they have a lot of different shapes. Some of them are kind of pearled, like you're seeing there, and others have this sort of undulating morphology. So we thought, what can be going on here? So one of the first things we wanted to do to explore this phenomenon was to look at other proteins. Is this some, very specific artifact associated with FUS, or is it more general? So we looked at two other proteins here. One is similar to FUS. It's the HNRMPA2, that low complexity domain of HNRMPA2, another RNA binding protein like FUS that is sort of hydrophobic in character and therefore tends to salt out of solution. But then we also looked at this RGG domain of LAF1, which, as the name implies, has a lot of charged residues, and it comes together more through electrostatic interactions and therefore has an opposite behavior with salt. So you can see over here, if we raise the salt concentration, I've already told you we see more and more tubules and more phase separation of the FUS protein. The RGG is just the opposite. As we raise the salt concentration, we screen the interactions that are driving the proteins together. So we see less and less phase separation, less and less tubules. So kind of the takeaway from this is these tubules, however they're forming, they seem to be somewhat general phenomenon with a variety of different kinds of condensate forming proteins. When these proteins assemble on membranes, they seem to drive the formation of these inward tubules. Okay, so what can be the mechanism here? So this is something we're still exploring, but I'll share with you our initial thoughts. So imagine a disordered protein floating in solution. It wants to form condensates. Two proteins come together, they can interact from many different angles. Then you take them and tether them to the membrane surface. Now they can't interact from very many orientations, right? Their interaction is really limited. But if they bend the membrane, they gain interactions with one another. And in that way, the increased interaction energy of the protein as a function of curvature can be used as a force to drive membrane curvature. So in effect, what we think is this protein layer creates a compressive stress on the membrane surface that drives bending of the membrane. Let's see, there might be a comment here in the chat. Let me see what's going on in the chat. Aha. Newt asks, what's the phase transition temperature for the RGG coacervates? Okay, this is a function of salt concentration, of course, but it can be tuned to be, in an accessible range, not far from you can tune it to exist between maybe 20 Celsius and 40 Celsius, so an accessible range. I think for the experiments we're doing, we easily see it phase separate at room temperature, so that the transition temperature must be above room temperature. Later in this talk, I'll show you another protein where we really carefully characterize the transition temperature. Yeah, good question. Okay, so we think we have this coacervate that's forming a compressive, or is driving a compressive stress on the membrane. So to try to understand this better, we collaborated with Padmini Rangamani. I don't know if she has participated in these symposia. If not, she might be a good candidate. She is a theorist who is interested, in many different kinds of modeling, but really has a strong history of doing continuum scale modeling of membranes. So she took a continuum scale modeling approach where she had a fluid membrane and she couples to it this fluid layer that is applying a compressive stress to the membrane. And with that modeling approach, she was able to recapitulate a lot of the different geometries that we had seen. And her model really makes a couple of predictions. It says that as you make the membrane more rigid, you ought to see more and more curved tubules, smaller and smaller diameters. Or sorry, other way around. As you make the membrane more rigid, the tubules should be less and less curved, right? Because the membrane will resist the work that the protein is trying to do on it as the membrane becomes more rigid. And then the second prediction is that as the protein layer becomes more rigid, you'll see more and more of these purled structures, because the purled structure has curvature in 2 dimensions. It's sort of a higher degree of curvature than a cylindrical tubule. So the more the protein is rigid, the more it will be able to drive the membrane into these purled morphologies. So we tested Padmini's predictions as we varied the lipid composition, gradually introduced saturated lipids, sterols, to drive the rigidity of the membrane up. And indeed, we see lower and lower curvature when you do that. And then as we increase the salt concentration to make this Fus protein phase separate more strongly, make the protein layer more rigid, we see more and more purled tubules. Let's see, I think another question is coming from Tim. Do you know what is the membrane composition constraint on this system? What membranes are you using now? I think your question, Tim, was probably asked on the previous slide, but is answered here. You can see the different compositions we have used here. So we started, the initial experiments I showed you were with DOPC, but then to test this prediction from Padmini, we explored a great range of compositions to increase the rigidity of the membrane. Okay. All right, so at this point, we've sort of established this idea that if you have disordered proteins that repel each other and you crowd them together on a membrane, they generate a steric pressure that drives convex bending. Then the converse is true. If you have instead disordered proteins that attract one another, you'll get concave bending. So next, let's consider what happens if you make some chimera that has both attractive and repulsive interactions. So here for the attractive domain, we're using FusLC, which I already introduced. Then for the repulsive domain, we took this domain from the protein AP180, which is a protein in endocytosis, and it has a very high net negative charge, which causes it to be quite repulsive toward copies of itself rather than forming condensates. So if you take this chimera and apply it to the membrane surface, you might expect to have attractive interactions close to the membrane, repulsive interactions further away. That should set up like this kind of bending moment. I teach undergrad mechanics, so I have all these props. Hopefully you can see this. So this kind of bending moment. Okay, and that would lead you to expect outward tubules, which is indeed what we see. Then you can take this chimera and turn it around, put the his tag on the other end and stick it to the membrane from the other end. Then you ought to have repulsive interactions close to the membrane, attractive interactions closer to the membrane, which should reverse the sign of the bending moment, right? Which should give you inward bending, which is indeed what we see. So sort of the takeaway message from this part of the talk is that this is these disordered domains seem to be able to generate all the curvatures and complexity that we find in the cell. And really, we can think of them as like a tool in the future that we could engineer proteins and control the shape of membranes. So from the way of thinking about building cells, this is where we're going with this work. Okay, so next, let's shift gears and talk about how can some of these ideas be important potentially in membrane organization in actual cellular processes. So to discuss that, let's come back to this picture of all the curvature in the cell. And now we are going to talk about one very specific pathway, which is the pathway of clathrin-mediated endocytosis. Now, in our laboratory, we really love this pathway. We have this saying that there are 3 certainties in life: death, taxes, and clathrin-mediated endocytosis. That may sound ridiculous to you, but if you think about how important the clathrin pathway really is, you'll understand better. So during the time, this 1-hour lecture we are having now, the surfaces of most of your cells will be completely recycled by clathrin-mediated endocytosis. Furthermore, it's this pathway through which all our envelope viruses, SARS-CoV-2, influenza, HIV, are internalized. And it's also the pathway we most frequently target to deliver therapeutics. So those are some of the reasons why we're so excited about this pathway. So before I can tell you what we've been doing, I have to share with you a little bit of background. excuse me, for the cough. So clathrin-mediated endocytosis takes place in several stages. The first stage is the initiation stage. So here you have a set of proteins that come together. They mark the site where endocytosis is going to happen, and they recruit the downstream proteins. But interestingly, they're not actually consumed. They remain at the plasma membrane and catalyze multiple rounds of endocytosis. So we think of them as catalysts. So once the catalysts are there, they recruit two types of proteins. They recruit adapters, and then the adapters ultimately recruit clathrin. And clathrin is this hexagonal coat protein that forms this soccer ball-like lattice. So once the adapters and clathrin are there, they work together to bend the membrane and generate these vesicles that are then internalized into the cell. Excuse me. Okay, so we were very excited to think about how to apply this sort of reverse engineering framework to this pathway. But it's kind of daunting because there are 3-4 dozen proteins involved in this pathway. So how do you tackle this problem? So we thought, let's start at the beginning with those catalyst proteins because there's relatively few of them. So this is the work of a wonderful postdoc, Casey Day, and graduate student Grace Cogo. So Casey and Grace, they got interested in these two proteins, EPS15 and FCHO. Both of these proteins form dimers and both have a high degree of intrinsic disorder. So you can see EPS15 here. It binds the membrane. In particular, it binds PI45P2 lipids, which is the main PIP lipid in endocytic vesicles. And it forms a dimer through this coiled-coil domain. And then it has this intrinsically disordered C terminus, which binds the structured mu-HD domain of FCHO. FCHO forms a dimer through its F-bar domain, which also binds PI45P2, and then has this intrinsically disordered middle domain. Okay, so these two proteins together form a network that has been known for years from these publications. However, the nature of that network and how exactly it is useful as a catalyst in endocytosis is not clear. So Grace and Casey, the first thing they did was to purify both of these proteins in full length, and they applied them to the surfaces of these giant vesicles, which contain PI45P2. So either the proteins alone, they just sort of coat the vesicle surface. They don't do anything particularly interesting. But if you apply both of them together, they form, interestingly, these condensates that are protein rich, leaving the surrounding membrane depleted of protein. And if you can control the sort of the faithfulness of this network by making mutations that affect the integrity, the ability of the proteins to bind each other. For example, if you remove this coiled-coil domain, that makes EPS15 into a monomer, you'll greatly reduce the frequency with which you see this condensation phenomenon. Also, if you make mutations to this interaction between EPS15 and FCHO, that also reduces the ability to see the network formation. Now, to kind of test what was going on here, we removed the membrane from the system, concentrate the protein, and look at the protein itself. And you can see these protein droplets sort of raining down onto the cover slip. Let me take a pause here. I just noticed there's some questions in the chat, which I wasn't paying attention. Sorry. Let's see. Hussein asks, have you investigated what determines the size of the tubules or if they can be tuned? Okay, that's going back to the previous part. So yeah, the size of the tubules, we don't exactly know what controls it. It seems if the protein layer gets too rigid, the tubules actually become larger in diameter, which is interesting. But we have found that, for example, the RGG protein makes substantially smaller tubules than FUS. So there does seem to be some difference from one protein to the next, but we don't fully understand it yet. And then Andres is asking, if you vary the amount of protein attachment to lipids, does it gradually impact the curvature? Can condensates bend the membrane even without attachment? Okay, I'll answer the second question first. So yeah, there have been a couple of labs. Let's see, I think the lab of Rumiana Demova is one, but I think I'm forgetting another one, who have shown that if you have droplets that are not connected to the membrane, they can sort of wet the membrane and the membrane can kind of wrap around the droplet. So then rather than having a tubule, you have like sort of a bud that's full of a condensate. But yeah, that has been observed. That's another way to bend the membrane with condensates. And then if you gradually add more protein, yeah, in general, if you have more protein, you get more tubules per vesicle. But in terms of varying the curvature, we haven't seen as directly an impact of the concentration of protein. But if you change things that affect how much the protein phase separates, like salt or pH, that can vary the curvature. Okay, great. All right, so coming back to the picture here, this EPS15 and FCHO seem to be forming these liquid-like condensates. Now here we're looking in the absence of the membrane, but the protein looks like it's forming these liquid condensates. So we thought we would characterize them a little bit thermodynamically. And I promised you some phase behavior. So here is where you will see it. So you can heat and cool this solution of protein. As you heat it, the droplets will dissolve, and then you can cool it back down and they will appear again. So you can do this heating and cooling. And what makes this useful is at any given temperature, you can take a picture of the solution. And if you carefully measure and correct for the background, then the concentration, or sorry, the brightness, fluorescence intensity within the droplets and the fluorescence intensity outside, once you've subtracted away noise and all that, those two intensities are proportional to the concentrations, roughly speaking, at the ends of a tie line on a temperature concentration phase diagram. So as you raise the temperature, the ends of the tie line are going to get closer and closer together until you reach the critical temperature. So here is such a phase diagram for EPS15 on its own. And you'll see that the critical temperature is below physiological temperature, sort of suggesting EPS15 may not form a stable network on its own. But then as you add a bit of FCHO and a bit more of FCHO, you can rapidly increase the critical temperature, which really illustrates that EPS15 and FCHO have this mutually supportive ability to form a network. Okay, but how does all this relate to endocytosis in the cell? So we needed a tool we could take into cells to probe this phenomenon. And here's where Casey, the postdoc working on this, had a really nice idea. Casey said, remember our mutation study. The most severe mutation was to make a monomer out of EPS15 by deleting the coiled domain. This monomeric version of EPS15 never formed any condensates, no matter what we did to it. So she said, let's insert into that monomeric form a light-activated multimerization domain. So she put in this Cry2 domain that is will multimerize when you shine blue light on it in a reversible way. And this tool, by the way, has been utilized by the Brangwyn lab and several other labs. So Casey adopted this from those labs. And so she made this solution of protein that when we shine blue light on it, will spontaneously form condensates. So that turned out to be a nice tool for working in live cells. Okay, so here, what you're looking at is a turf microscopy video of the surface, the adherent surface of a breast tumor epithelial cell. And in this cell, there you see all these tiny little dots, and each one of these dots, if you watch it, it will show up and sort of get brighter and linger around for a little while and then eventually depart. And that whole process takes about a minute on average. So a number of labs have studied, so each of these dots is an endocytic event, and I should say they're labeled in this movie by two proteins. EPS15 is labeled, and also AP2 is labeled. AP2 is the most abundant adapter protein of clathrin-mediated endocytic vesicles. And so it's sort of our marker of where endocytosis is taking place, and it's recruited by EPS15. So a number of labs have studied the dynamics of endocytosis, and they have found something interesting. So most of the time, these little dots lead to a productive endocytic event where we create a vesicle. But now and then, you get proteins that assemble at the membrane and just stochastically disassemble without ever creating a vesicle. We call that an abortive event. And detailed work in this area has revealed that an event that lasts longer than 20 seconds is usually abortive. Then at the other end of the spectrum, sometimes you have these like super stable events. They also tend to be large or therefore bright in our images. And they don't result in endocytosis. They just linger at the plasma membrane. They may be important in signaling or autophagy, but they don't seem to be endocytic structures. Okay, so in these cells, here's the distribution before we started manipulating the cell. So in wild type cells, most events are productive. A few are abortive. Stalls are quite rare. Then if we knock out EPS15 using CRISPR, we about double the fraction of abortive structures, which kind of makes sense. You're messing up the catalyst, so abortive structures become more common. Then if you replace EPS15 with EPS15 Cry2, but you're careful to keep the light off, so that means EPS15 is acting like a monomer, then you have the same defect, this increase in abortive structures. But then if you gradually apply blue light, and you can find a level where you basically get back the wild type behavior, so that the extent of multimerization is sort of similar to a dimer, presumably. Now then, if you keep increasing the light, you might think, oh, we'll drive the abortive structures down more and more and make sort of like super productive endocytosis. It turns out that's not what happens. So here we're increasing the blue light about 5 to 10-fold. You can see the abortive structures barely budge, but you see this big like 4-fold increase in stalled structures, which was super weird. So you can see it in this video. See all these really bright structures that don't go anywhere. Those are the abortive structures, or sorry, these stalled structures. So we were really confused by that. So to try to understand, we went back to the purified protein droplets, and we looked at droplets formed with low light versus this strong light. And you can see here, the ones formed with low light, they fuse right away. They're very liquid. But the ones formed with strong light, you get like just this sticking, and these two droplets stick together, and you never see this rerounding, even after many minutes. And the FRAP bears that out too. The structures exposed to low light recover really similarly to the wild type protein droplets, but the structures exposed to strong light recover much more slowly. So then if you take the same FRAP tool and you apply it to the cells themselves, you can FRAP the endocytic structures and watch them recover. So here we're frapping one endocytic structure that's expressing EPS15, and you can see the time scale of EPS15 recovery. And if you average a bunch of those structures together, this is the recovery curve. So then if you replace EPS15 with EPS15 Cry2, but you keep the light off, you get actually a faster recovery, suggesting that when EPS15 is a monomer, it's not stably recruited. It's coming and going from the endocytic sites really fast. Then if you apply this low level of light that gave us the wild type dynamics, you get back a wild type rate of recovery, really suggesting that this protein, its stability at endocytic structures is very similar to the wild type protein. But then if you apply this really high level of light, you see a slow level of recovery, suggesting that with a high level of light, these complexes formed by Cry2-EPS15 are too stable at the membrane. They're not exchanging and maybe not allowing the membrane to rearrange. Interestingly, this behavior is passed down to AP2, which is the next adapter protein. And it really shows that even though we haven't done anything to AP2, the dynamics of the catalyst, EPS15, ultimately determines the dynamics of AP2 and therefore the whole vesicle. So kind of the conclusion from this part is twofold. All catalysts have two jobs, right? If we just think back to undergrad chemistry and think about catalysts, a catalyst has to bring together reactants and it has to release products. So in this case, the reactants are all the protein components of the endocytic vesicle. And if the catalyst is networked into a loose network, that helps it in recruiting the downstream components. But then if that network becomes too stable, now it becomes harder to complete that second function, which is to release the product. And in this case, the product is like the finished endocytic vesicle. So if the catalyst network ultimately has to be flexible enough to allow the vesicle to mature and depart. And we think maybe a liquid-like network is ideal for that catalytic function. Okay, so that is the conclusion of this second part. Let me check and see. Okay, yeah, no new questions. All right, so far, hopefully I've convinced you that endocytosis is catalyzed by a flexible protein network. And in this last bit, we're going to ask, is there any way to use that understanding to promote therapeutic delivery? And this question is motivated by the fact that many therapeutic carriers, nanoparticles designed for uptake by cells, most of them target the clathrin pathway because after all, the clathrin pathway recycles most receptors. So to ask this question, this is the work of Grant Ashby, a graduate student in our lab. And Grant decided to adopt this turf microscopy framework that we have in the lab and try to use it to investigate this question. So he created a set of model particles. These particles are really simplistic. They're just biotinylated vesicles with an average diameter of about 50 nanometers. And in this movie, they're colored red. And then the endocytic machinery here is actually colored deep red, but I put it blue in this movie to make it easier to see. So you see in this movie, two sets of dots, blue and red. And now and then the dots colocalize and a vesicle is taken into the cell. So you can see such an event here. The vesicle is there in red, endocytic machinery comes along and assembles and takes the vesicle into the cell. So we looked at tens of thousands of these events, and I'll tell you what we learned about them. Excuse me. Okay, so the first most obvious thing that we saw is that if you compare targeted vesicles, which have biotin, to untargeted vesicles, which don't have biotin, the targeted vesicles do a much better job of finding their way under the cell. So here we're looking at cells that are adhered to a cover slip because we're in turf microscopy. So to get under the cell, a vesicle has to diffuse down there. Then it has to navigate through all the extracellular matrix that the cell is secreting and between all the focal adhesions and find an endocytic site. So this is a difficult task. And it looks like targeting, perhaps not surprisingly, helps vesicles get under the cell. Some quantitative evidence of that is as follows. So here is the initial distribution of vesicle sizes. Then if you look at the size distribution of non-targeted vesicles that make it under the cell, you see they're substantially smaller. The average diameter shift from like 50 nanometers over to about 30 nanometers. But then if you look at targeted vesicles, they better mirror the initial size distribution. Okay, so that's the first observation. Targeting helps navigate the extracellular matrix, navigate between cells, and get under the cell. Then the second observation has to do with dynamics. Excuse me 1 minute. Need another cough drop. Okay, so here's the dynamics of the wild type cells without any vesicles applied. Then if you look at events or look at vesicles that take up untargeted vesicles, you see two things. One is that there seems to be some stability effect. So perhaps it's the curvature of the vesicle, but we see fewer abortive events when we have vesicles there, even if they're not targeted. The other effect is that we see more stalls because the vesicle itself is a mechanical barrier to uptake. It's something that endocytic vesicle has to internalize. And both of these effects are somewhat exacerbated, especially the stalling effect, by targeting moieties, by having biotin on the surface of the vesicle, perhaps because then we can bind much more strongly to the membrane surface. And ultimately, rather frustratingly, targeting doesn't increase the fraction of successful uptake events very much at all. And we think this is for two reasons, or really there's two sides to this. So targeting does promote vesicles getting under the cell, but then ultimately those vesicles that are now very much stuck to the cell surface by binding to receptors are presenting a greater mechanical barrier to uptake and are, even though they're under the cell, are ultimately harder to take up. So it's a bit of a can't have your cake and eat it too situation. And that's sort of the history of delivery through the endocytic pathway. As much as I love endocytosis, it's a very difficult thing to deliver therapeutics through endocytic pathways because it's hard to get the cell to take up the particles. And once the cell does take up the particles, oftentimes they are degraded or expelled. So in the very last bit here, I'll share with you something that's maybe in the theme of artificial cells that we are an area we're exploring to improve delivery to the cell cytosol. And this is to build vesicles that deliver therapeutics into the cytosol using the gap junction network rather than the endocytic network. So the gap junction network, if you're not aware, is a network of hexameric pores that connect the cytosol of neighboring cells in most tissues. Gap junction consists of this protein called connexin. When a hexamer of connexin on the surface of one cell finds another cell, a channel is opened and small molecules, metabolites, many small molecule drugs, linear molecules like peptides and single-strand nucleotides can move from one cell to the next through these channels. So our idea was to make vesicles that incorporate these connexin channels and use them to release cargoes into the cytosol. This is the work of three great women in our lab, Avi, Amy, and Andrea. So when they started out this work, the first task was to find a way to make these vesicles that contain connexin proteins. And the strategy they took was to overexpress connexin in living cells and then extract the membrane of these living cells through the process of blebbing. Some of you guys are probably familiar with blebbing. So then once they had these blebs, they verified that indeed the connexins are functional. And the way to test that, or a simple way to test that, is to look at the sensitivity to calcium. So calcium closes gap junction channels. And here you can see when calcium is present, dye is retained. And when calcium is removed, dye is released. Then if you apply these vesicles to the surfaces of cells, you can see that when they settle on the cells, they quickly transfer some of the molecules within them into the cytosol of the cell. If you load them with a drug like doxorubicin here, a very classic chemotherapeutic drug, you can look at the effect of this delivery through these connexin vesicles on the lethal dose of doxorubicin. And here we were excited to see that using a connexin vesicle, rather than just delivering the drug directly to the cells, reduces the lethal dose of doxorubicin by almost 100-fold, which is quite surprising and exciting. We've also looked at this approach for in vivo delivery, in particular intratumoral injection into breast tumors. And there we see that these particles do a much better job than conventional liposomal doxorubicin at getting the drug to its site of action, which is the nuclei of the cells. Okay, and future work in this area involves several collaborators. The first is Brian Bellardi, who's in the theme of the synthetic cell. He is making these particles recombinantly using purified lipids and proteins and engineering in some exciting chemically and light activated functionality. And then we're also working with Janet Zolden's lab, who's interested in using these particles to reinforce the junctions between cardiac cells and artificial tissues. And also Hugh Smyth's lab, who's using these particles to repair asthmatic epithelia in a lung model. Okay, so I'm going to stop there. That was a lot of stuff, and I'd be very happy to hear your questions. We can start with the ones in the chat. So from Tanner, we have, do you see any relationship between the number of stalling events and the concentration of, oh wait, okay, something new showed up, which pushed that off. Stalling events and the concentration of biotins on the vesicle membrane? Good question, Tanner. So what I showed you was no biotin and 20% biotin, which is quite a lot. We also tested 10% biotin. Interestingly, 10% so far seems to be a little bit of a sweet spot where we get fewer stalls and more uptake. But I didn't include that because we need more data on that. We're not sure if that is true, but it may be the case that there's some optimum degree of targeting, which would be exciting. Then Stefan says, would it be possible to engineer an artificial gap junction between two vesicles without a live cell at all? Yes, quite possibly. Yeah, all you need to form a gap junction is the connexin itself. So like Brian, he has already done this. He can see two vesicles bind one another and form junctions with each other and transfer dye between vesicles. Then Aditya, how do you think the endocytic initiator assemblies might be influencing the membrane itself? Aditya, do you mean like the mechanical properties of the membrane or what aspect of the membrane?
Yeah, I mean, for example, domain formation on the membranes or just the phase behavior of the membrane itself.
Yeah, that's an interesting thing. So, okay, in the history of the clathrin field has not been very friendly toward the lipid raft field. But putting those politics aside, it's generally thought that lipids like PIP2, which in the cell are quite unsaturated, that they may not mix well with lipid rafts. And similarly, a lot of, like you probably know the work of Ilya Leventhal, right? Ilya has shown that some of our very favorite receptors that really go strongly into coated vesicles, like like transferrin receptor, that those are also receptors that avoid lipid rafts. So it may be that endocytic sites are sort of like non-rafts, you know what I mean? So yeah, I think there is a phase preference. It's likely that when those initiators assemble, that they may create, like, they may exclude cholesterol or sphingomyelin. That's possible, but we don't really know. One of the reasons we know so little, you're probably aware, is that in live cells, it's such a difficulty to label lipids. If you label a lipid, you change its behavior. And so it's really hard to know, like, I think still today, this question, are endocytic sites, are they sort of synonymous with RAFs or are they anti-RAFs? It's really not clear.
Thank you.
Sure, thank you. Okay, so Peter says, for the liposomes you inject to tumors, are they pegylated? Yes, they are. Or wait, no, they are not. They are not. That's right. I'm thinking of a different project because these are cell-derived particles, so they are not pegylated. And then Taisuk, to make liposomes better for cargo delivery, do you have any advice and insights? Okay, make them as small as you possibly can, remembering that the clathrin-coated vesicle has an average diameter of only about 50 nanometers, which really makes it delivery a challenge. So that's the first thing, make it small. And then I would just say, think carefully about targeting. There can be such a thing as too much of a good thing. Remember that a particle that sticks really strongly to the plasma membrane may present a mechanical barrier, even though it's concentrating receptors, which is kind of counterintuitive. Then Ava, Has anyone reconstituted clathrin-coated pit in vitro? So people are constantly working on that. So the most impressive advance in that direction that I saw, what is the name of that guy? I will think of it. Oh, there's a lab in Europe where they were able to recruit, use an adapter protein, recruit clathrin and make buds on the membrane, and then actually recruit dynamin and cut them off. So that's the closest I've seen to a full reconstitution. And yeah, let me think if I can remember the name of the guy in Europe that did that. Unfortunately, he passed away. Yeah, okay. While I think of that, any other question?
I just want to say thank you so much for an excellent talk. This is really interesting. Seeing those, seeing those membrane deformations, it just makes me think I want to have cell division, and I'm really waiting for that. So I have a question for the RGG part of, do you see that if the RGG domains aggregate, do you see any sequestration of RNA? Or do you have anything inside those vesicles? Like if you have protein expression system, we and other people have seen that those RGG coacervates, they really soap up tRNA and like they kill cell extract. Do you see that too, or are yours better behaved?
So this is, we purify RGG from bacteria prior to the experiment, right? So, so we don't have an extract present in our experiments. I mean, that being said, that some RNA has come along in the purification is quite possible. We kind of follow the protocols from other groups, but I know some of the groups, especially those like doing NMR or doing really detailed studies on RGG, will do some additional testing to make sure there's absolutely no nucleotide content, and we have not done that. So there could be some element of that in our results. For sure. But yeah, if we were to do this in an extract, or if we were to on purpose add some RNA, for sure, I would expect it to also get recruited to the membrane, and that could be interesting too.
Yeah, that would be very cool. All right. If there are no more questions, I want to say thank you so much again, and thanks everyone else for a great discussion.
Yeah, thanks for all the great questions. It was fun.
And have a great rest of your week.
You too. Thanks. Bye.
Bye.